Welcome to everybody. My name is Carolina and I'm hosting the seminar of today. It's really a pleasure for me to introduce Laura Valle. So just to give a very brief overview on her career, she got her PhD in Madrid at the Spanish Cancer Center, and later on she moved in US in Ohio for her postdoctoral training with Albert de la Chapelle. And I think there she started focusing on a bit more on her topic, on her actual topic, that is colon rectal cancer. And she moved back to Spain and to IDIBELL directly in 2009. And then she started her own group on hereditary cancers, which is, I would say, a very fascinating topic. I think you all are aware that there are some cancers that have a genetic predisposition, and Laura is particularly focusing on colorectal cancer, so intestinal cancers, and hereditary and genetic predisposition to cancer development. I have to say that Laura is still in a wonderful career path. I want to remember a few of the prizes and the awards she was winning in the last years. Among them, the L'Oréal Women in Science, that is a very prestigious award recognizing women in their scientific career, but in general, scientists. She got also a very outstanding prize from the Human Genetic Association, and is a national award that recognizes young researchers. And this is also a very important prize. She had lots of publication on her topic, and she is really forefront in this field of research. And so I leave her to her presentation. She will talk today about understanding the cancer-predisposing syndrome caused by defective proofreading activity of polymerases, eta and delta. So thank you, Laura, and please, I leave you the stage.
Thank you, Carolina, for the nice introduction. Before I begin my talk, I would like to thank you all for being virtually here. I hope you are all in good health, both mental and physical. And without further delay, I will begin this seminar on the cancer-predisposing syndrome caused by defective proofreading activity of polymerases epsilon and delta. So it was in 2013 when this syndrome was first described by the group of Ian Tomlinson. And from there, these past 7 years, we and others have tried to better characterize the syndrome, both from the clinical and molecular perspectives. For today, I have prepared this presentation where I will talk about genetics, the clinical characteristics, and tumor molecular characteristics. And then I will also talk on something we've been working these past times, on variance interpretation and classification, and also on one topic that is quite challenging, but a matter of discussion since the description of the syndrome, which is what happens with the loss of function variants and variants outside the exonuclease domain. And finally, very briefly, some notes on prognosis and immunotherapy. So the polymerase proofreading associated polyposits, that from now on I will call PPAP, is a cancer predisposing syndrome inherited in an autosomal dominant manner. It is caused by germline pathogenic variants that alter the proofreading activity of polymerases epsilon and delta. In particular, these genes or these polymerases that code for the epsilon and delta polymerases are very special polymerases. We might call them high fidelity ones because while they replicate the DNA, they are able to correct their own errors thanks to this proofreading activity that they have. They have this exonuclease activity that while they replicate the DNA, they are able to eliminate the errors that they introduce themselves. So when there is a mismatch repair, we know that the Lynch syndrome is caused. This is a mainly colorectal cancer predisposing syndrome. But when there is a problem in the proofreading activity of these two polymerases, we have this Ppap syndrome. We may find genetic variants along the whole sequence of these two genes. In fact, these are relatively large genes. However, only those affecting the exonuclease domain are the ones inequivocally linked to this syndrome, to this Ppap. In this table, I have introduced some of the best known reported Ppap-associated variants, and the most recurrent is this one, L424V, that has been found in numerous families all over the world. They all affect the exonuclease domain, as I have already said. And what is important is that they all, or almost all, are in direct contact to the DNA that is being replicated. They are in direct contact or affecting the DNA binding pocket. I mean, where the DNA is there with the polymerase and exonuclease. So this is something important that we will take into account when I will explain the variant interpretation. So as most of you may know, proofreading mutations are also found in tumors, not only in the germline. Okay, so this is particularly relevant for POLE. POLE exonuclease domain mutations are identified in 7 to 12% of endometrial cancers and in 2.5 to 3% of colorectal tumors. And these are the most common ones, but then we found these mutations in other tumors, such as tumors of the brain, ovary, pancreas, stomach, breast, kidney, bladder, or prostate. However, somatic POLD1 exonuclease domain mutations are extremely rare. We rarely found them in tumors. And finally, just to highlight, because this is important for Ppap, that somatic and germline mutations are usually different. So somatic hotspots are very, very rarely found mutated in the germline, and the other way around. Moving on to the clinical characteristics, considering the variants that have been classified as pathogenic or likely pathogenic by our group or others, we know that the most frequent phenotype is the colorectal cancer, diagnosed at a mean age of 35 to 50 years. And also, we usually find adenomas, an attenuated adenomatous polyposis phenotype. In addition to this, other tumors that are quite frequent in these patients are endometrial tumors, ovarian, and breast cancer. However, since we can call it a DNA repair defect, other tumors have been observed in mutation carriers. At the upper gastrointestinal tract, there we can find also adenomas or malignant tumors, tumors in the brain, small intestine, pancreas, and others. And I will talk about this later, but there is a very special phenotype that is called constitutional mismatch repair deficient-like phenotype that occurs, because only 2 reports are from this, when somatic hotspots occur in the germline. But this is, as I said before, very rare, but I want you to know about it because it's quite interesting. Here, I wanted to show you a typical pedigree from one of these families. This plus means they carry this POLD1 mutation. It's a pathogenic mutation in the gene. And as you can see, we have several members of the family diagnosed with colorectal cancer at different ages, but it's usually in the adult age. But we also see polyps, endometrial cancer, upper gastrointestinal phenotypes. So this is typical. But then we see these specific phenotypes in people carrying very rare mutations in the germline, that usually are recurrent as a somatic hotspot in tumors. So as I've said before, somatic mutations we find in tumors are rarely found in the germline, and vice versa. So here I want to present the two cases that have been reported in the literature. One is this individual with a POLE V411L mutation. This is one of the recurrent somatic hotspots. And when this person had this mutation in the germline, it was a 14-year-old male that had already developed the colorectal cancer. Moreover, he also had the typical polyps that we find in the syndrome, these adenomas in the colon and other parts of the gastrointestinal tract. But very curiously, these carriers have what we call CALMs, that is café au lait macules. These ones that you can see in these patients that are typical from neurofibromatosis or constitutional mismatch repair. And then pilomatricomas. And something different but similar for this other mutation that has been described in a 5-year-old child with a medulloblastoma. And he also had these hyperpigmented macules and papules. Some of them were CALMs, these café au lait macules, renal cyst, osteochondroma, and 3 pilomatricomas. So it seems these mutations are much more aggressive than the ones we usually see in regular PPAP patients. So until recently, most of the studies that had been published were focused on colorectal and polyposis cohorts. So most of the phenotypes were related to these phenotypes. However, looking at all the pedigrees that we see in the clinics, we realized that there are many other tumors that were present in these families. And that's why we decided to evaluate the prevalence of these variants among hereditary cancer patients in general, not focusing only on colorectal cancer patients. And to do that, our group has studied over 2,800 unrelated probants referred to our hereditary cancer program. We've analyzed those, and we now can estimate a prevalence of 0.1 to 0.4% of familial cancer cases carrying a pathogenic or likely pathogenic mutation in one of these two genes. When we focus on colorectal cancer and polyposis patients, this percentage raises a bit to 0.3 or 0.7%. So what does this mean? No, this means this is a relatively rare syndrome, but as the consequences for the patients are quite relevant, we believe it's important to study it. And indeed, it's already for years has been included in multi-gene panels for genetic diagnostics. So regarding the clinical management and surveillance, in 2016, we made an attempt to provide the clinicians with some preliminary recommendations for clinical surveillance, which basically took into account the gastrointestinal findings and highlighted the risk to other tumors, such as the endometrial tumor, breast cancer, and also we highlighted that brain tumors were also quite frequent in these families. With the data other groups and our group are currently gathering, we believe we will be able to update this surveillance proposal in the very near future. We move to one of the, in my opinion, one of the most interesting parts of the talk, where I will talk about the characteristics of the tumors. And as I introduced in the first slides, the major function of polymerases epsilon and delta is to replicate the genome. And already with that activity, the misincorporation of nucleotides by these polymerases is very low. Moreover, they have this proofreading function, which reduces the replication errors further, and they reduces these errors to further 100-fold. So whether these mutations are germline or somatic, what they cause is a very high mutation burden in the tumors. In this graph, what we see is the regular tumors, the ones that do not have a very high mutation burden, which can be divided in copy number low or high. But then we move, and what we've known till recent years is the next group of tumors with more mutations were the ones with a mismatch repair defect, or what we call microsatellite unstable tumors. And in this case, we can see that they have a very high mutation rate. These are the numbers of mutations by megabase. And we already see these already accumulate a lot of mutations. However, when we look at the mutated POLE or POLD1, which in this case, because these are somatic mutations, when we look at the POLE or POLD1 mutated tumors, what we observe is that this mutation rate is even higher. It's what we call ultra-mutated tumors. This is an example of endometrial tumors, but this happens in colon or other tumor types where polymutations occur. So next, I will briefly explain the concept of mutational signatures. Mutational signatures are characteristic combinations of mutation types arising from specific mutagenesis processes. So basically, mutagenic processes leave a footprint in our DNA. And here, this has been tested in vitro. These are induced human pluripotent stem cells exposed to mutagens, and when they observe, for example, when they simulate the sunlight and expose these cells to this mutagen, they observe that the cells accumulate one specific type of mutation, CT, in a specific trinucleotide context. And the same when they expose the cells to something similar to the tobacco smoke. And in this case, you observe there are different types of mutations accumulated. When we look for these patterns in the tumors from the cosmic database, we observe that this corresponds to signature 7, and there are tumors that have the same profile, and the same here for signature 4. In fact, we observed that melanoma accumulates signature 7, which is the one that was associated with the sunlight, while lung adenocarcinoma accumulate this signature 4. However, mutational signatures do not only represent the external exposure to mutagens, but we can also find specific mutational signatures when there is a defective DNA repair, either in the tumor or in the germline, but that causes this tumor. And here in this graph that is taken from this paper from Alexandrov, we see the different mutational signatures that are formed by characteristic mutations in a specific context, and here the different tumor types. So if we look for specific mutational signatures for mismatch repair deficiency, we observe that there are these at least 4 signatures associated with this DNA repair deficiency. But we see the same for base excision repair mut-Wiyage mutations or NTHL1. So what happens when we look for specific mutations or mutational signatures for POLE? So there is a very, very specific one that is signature 10. There is really specific for this alteration. Okay, we don't see it anywhere else, only when there is a mutation in POLE or POLD1. And as you can see, the tumor types that are commonly gathering these mutations, or these mutational signatures, are colorectal cancer and endometrial cancer. But signature 10 is not the only one that we see in these tumors, because sometimes when a POLE mutation occurs or a POLD1 mutation, due to this high error-prone mutation rate, these mutations can target a mismatch repair gene. And in this case, we have a combination of mismatch repair deficiency and a POLE mutation or POLD1 mutation. So what happens in those cases? In these cases, we have a specific signature. We have a signature that combines POLE and mismatch repair deficiency, which is signature 14, and another one that recapitulates a POLD1 mutation together with mismatch repair deficiency, or MSI, which is signature 20. So basically what we know is that POLE and POLD1 mutated tumors, whether they are caused by a somatic or germline exonuclease domain mutation, they accumulate lots of mutations. They can over 10,000 mutations per megabase, and very usually over 1,000 mutations per megabase. And they have this specific mutational signature 10, which corresponds to C2A transversions in the context of TCT, and C to T transitions in the context of TCG. Okay, so we have recently made the effort to define gene-specific, well, this is something we've been working on for a long time, I'd say. And we've made the effort to define gene-specific ACMG-AMPC-based variant classification guidelines for these two genes. And basically what we do is gather the evidence available on population allele frequency, missense, and the variant has to be a missense variant and located in the sequence coding the exonuclease domain. We also take into account segregation data, functional studies supporting a mutator phenotype in GIST, and finally, the presence of a high mutation rate and the mutational signature 10, or in the case of MSI tumors, the high mutation rate together with the presence of signature 14 or 20, depending on the mutated gene. So, as I've just told you, we have attempting to define these specific classification guidelines for the genes, because these are quite unique genes because of this thing that the mutation has to be at the exonuclease domain and they cause this accumulation of errors. And basically we take into account what I've just said, and we have paid special attention to the location of the variants within not only the exonuclease domain, but within the DNA cleft, where the DNA gets in contact to the polymerase. Okay. So this is something we have very strongly studied and considered. So one problematic issue has always present when talking about Ppap or somatic POLE and POLD1 mutations is the topic of loss of function and outside the exonuclease domain variants. I want to make sure everybody understands that so far, the only variants unequivocally linked to Ppap are those missense affecting the exonuclease. This is very important because we are used to something different. So we have studied this, and I will now show you some data that corroborates this. But basically, I want you to know that loss of function and outside the exonuclease domain variants do not cause Ppap or colorectal cancer predisposition. So we've taken a look into the frequency of loss of function variants in familial cancer, and within this group, familial colorectal cancer and polyposis, and compare these frequencies with the controls. And we've observed very similar frequencies in all three groups, and something similar occurs for outside the exonuclease domain. So we wanted to further study this because some people are reluctant to believe this. We performed whole exome sequencing in 4 tumors developed by loss of function variant carriers, and we have not found hypermutation or poly or associated signatures. Moreover, we have identified a lot of variants outside the exonuclease domain in these genes in our familial cancer cases, and we tried to find whether these variants were somatically mutated and in the tumors that are stored in the TCGA database. And when we analyzed these tumors that they had these variants outside the exonuclease domain, none of them had the POLE or POLD1 associated mutational signatures, none of them. And if some of the tumors that we studied had them, it's because they had a mutation, a real somatic mutation in the exonuclease domain of these genes. So everything points towards the fact that only the missense mutations affecting the exonuclease domain are causing this syndrome. In fact, it makes sense because Ppap, as its name refers to, is caused by aberrant polymerase proofreading activity. This means when you have a loss of function mutation in this gene, it's a heterozygous mutation. So one of the alleles is gone. It's probably gone, it's truncated. So you have the wild type, the healthy allele, working for replication and working for this proofreading activity. So there's no DNA repair problem here. So this is quite important. As I've already said, we are used to classify loss of function variants in non-cancer predisposing genes as likely pathogenic, but this is totally different. And as of today, only carefully curated exonuclease domain missense variants should be considered as potentially associated with Ppap. I'm getting to the final part of the talk. I want to briefly comment on the implications for prognosis and immune therapy of these mutations, whether they are identified in the germline or in tumors. So as we have previously mentioned, proofreading mutated tumors display very high mutation burden, which cause the production of a very high number of neoantigens. So this translates in the fact that these tumors show high density of tumor-infiltrating lymphocytes with high numbers of CD8 positive cells and enrichment of genes encoding immunosuppressive checkpoints such as PD-1 and PD-L1. This is as a consequence of this, we usually see a Crohn-like reaction. This has been linked to good prognosis, as you may probably already know. This has been the hypothesis that the anti-tumor immune response generated by the neoantigens is responsible for this good prognosis. And also, several articles have reported the clinical benefit of immune checkpoint inhibition for these tumors. This is the proposed mechanism linking polymerase exonuclease domain mutation, immune response, and favorable cancer prognosis. So this is the polymerase that is replicating the DNA, and while the exonuclease is mutated, is altered, the proofreading is not being performed correctly, and there is accumulation of mutations. This translates into the production of mutated neopeptides, which are presented by the MHC class 1 on the tumor cell. This translates into the checkpoint activation and increased T cell toxicity. This is an example of improved prognosis and survival for endometrial tumors. And as you can see, polyproofreading mutant tumors have an excellent prognosis and survival. So I'm going to finish with this. Thank you, because now I'm starting to lose my voice. I want to especially thank my team, and particularly Pilar Moore, who's done a lot of work on polymerase epsilon and delta 1 these past years. And I hope you can see our paper soon published, which recopilates a very interesting study on all the over 2,800 probants from the hereditary cancer program here at the Catalan Institute of Oncology and IDIBELL. And I want to thank all our clinical collaborators, even from our group or other groups in Spain and Catalonia, and also all the people helping us with the tumor whole exome sequencing analysis, the in silico structural analysis, GIST assays at the university. And thank you all our funders and the networks, the European networks we belong to. Thank you all for your attention. And now I will take questions. You have to raise your hand. I don't know, maybe Carolina, would you explain this now?
Yes. Thank you, Laura. Presentation, it was very clear. So everybody that is interested, please, in putting question, use the button function, raise your hands, and I will see you appearing, and I will give you possibility to answer your question. Remind to unmute your microphone. And maybe I start in the meantime asking you something. I was very intrigued by, so the mutation are in genes and protein that do actually have a very basic function for many different tissues and cells. Do you know why it is so prevalent, the tumor in the colon? Colorectal cancer is the main, and also the endometrium, no, you were saying. Why there is this special sensitivities?
We've known this for a long time because it is the typical tumor spectrum that we see in DNA repair associated syndrome. So when you're, for example, in Lynch syndrome, it's the same. You have colorectal cancer, endometrial cancer, ovarian. So it's more or less the same spectrum of tumors. And that's because they are involved in DNA repair. So these organs are highly exposed to very drastic, very strong damage, DNA damage. So you need a very, very good and very and well-working system, DNA repair system. Otherwise, you would get a tumor. Colon is highly exposed to this stress.
I mean, for example, also the skin and the blood are normally tissue that are quite exposed, but there is no real correlation with blood tumor and skin, like melanoma or other kind of skin cancer.
In fact, there are several reports on Ppap with melanoma cases. We are studying this, the variants that they have found very carefully, because most of them have not been yet classified as likely pathogenic. They remain as variants of unknown significance, but there is unlike Lynch syndrome, where you don't see skin tumors. For Ppap, it seems you may have some predisposition to skin tumors. But as I said, it's not totally demonstrated, because the variants that we find in these families have not been still classified as likely pathogenic. So we cannot say this.
Did you have a chance to check on the mutational signature and see whether it is prevalent, the melanoma associated or the one associated with POLD, POLE?
Yeah, we've studied multiple tumors now that come from Ppap patients, and we see the mutational signature is independent on the tumor type, I'd say. I mean, if you see POLE associated signature 10, you see it in breast, in ovarian. If it's caused by the syndrome, you detect it. We have problems to detect signature 10 when there is a secondary mismatch repair defect, because it seems this one sucks up all the signatures. But in this case, we look for the 14 or 20. But otherwise, it's and this has already been seen for example, for the NTHL1 associated tumor syndrome. No, many tumor types, and they all share the same mutational signature.
Very interesting. You were saying, I keep up talking. Please raise your hand if you have questions. I have probably the last one on my side about the immunotherapy. You said that now this tumor are having success based on immunotherapy, because this high frequency of mutation is working nice in raising up the response. But you are also pointing a lot the attention on this DNA binding part. Do you think there might be possibility to develop different therapeutic approach, knowing that this DNA binding is quite important for the disease?
Well, I don't know. We haven't looked into that, the truth. But it seems, I mean, they have very good prognosis in general. But anyways, it's if it's a very aggressive, I mean, we have brain tumors and things like that. So it seems immunotherapy could be the thing. If it works, that's okay. But it's true that so the thing is that this DNA binding thing has to do with the fact that if the residue that is involved in repairing that while the DNA is passing through is damaged, that's the problem. I mean, you can have a variant in the exonuclease domain that is a bit farther from DNA, and it may not do anything to the DNA repair. I mean, it might change a little bit the conformation and things like that, but it doesn't really affect that process. So whether it can be used for therapies or something, I have no idea, but it could be interesting. The thing is whether it is needed or not, because if the prognosis is good, and if it's there are doubts that it's going to be cured, and you use immunotherapy and it works, so we have to be aware whether this is really necessary or not. and that's important. But it's a good point. Thank you.
That was a great talk. I didn't know much about POLE and POLD. So those two mutations that they produce the CCMMR-like, they're great, right? So those are the ones in the DNA binding domain that they bind with more affinity to the DNA.
In fact, no. I mean, the thing is, for example, 411 is not in direct contact with the DNA, but it affects one of the residues that define the cleft, the DNA binding cleft. So it has. So what we look for is things that are in direct contact with the DNA, but also can affect this contact. So the ones that are defining this pocket where the DNA passes through the polymerase, it's not, in fact. It's one of the ones that is the few ones that are not in direct contact with it. So it has nothing to do with that. But what seems to us is that these somatic hotspots are quite aggressive, and I mean, this is just a theory and we don't know, but maybe most of them don't, they might be lethal in the, because we don't see them much. Only these two cases have been reported, and with these extremely aggressive phenotypes.
So I was going to ask you two things. One, at the level of the patient, right? So in those that they have hypermutated tumors, but you cannot find a germline mutation, have you checked if there are mosaics for these two? Because then if they're mosaic, but they're highly aggressive, they will be likely be very tolerated, right? Like happen with the typical mosaic aggressive mutations. And the other question was, you haven't checked that. Nobody has reported mosaics in those.
No, we have identified several de novo mutations, but not mosaics so far. But we haven't. There are not so many cases reported up to date. I mean, there could be with real pathogen. I mean, it's difficult to know whether they really exist, but they probably do, as in most syndromes. But I think we will discover those further on, because we are still in a. Even though we've been working on this for almost 7 years now, it's since it is so rare. I mean, I think we need more time to get to mosaics, but hopefully those will come up. But it's a good point that if they have aggressive phenotypes, they could be mosaics too.
Yeah, and then the other question was about the mutations itself. So the other copy at the tumor level is always there. It's still wild type. They don't have a load. It's more like a gain of function type of thing. Not exactly, but sort of. Haploinsufficiency,
I would say. I mean, the thing is that if you don't have the allele, the wild type has to be there. Otherwise, there's, one copy has to be there, and the other one has to be producing this incorrect correction of the replication. So that's the thing. I mean,
and has anyone done a pull down or like, does these two super aggressive compete for the binding? Because you still have the other copy. Probably, as you said, if you don't keep one copy, it's a catastrophe. The cell doesn't divide. Fine. But now, if the very aggressive one that they don't bind completely, do they compete with the wild type? And then it's sort of.
We haven't, and I mean, nobody has tested that. But it's a very good idea.
There are also a couple of questions from Marta. You were right. She has a problem with the microphone.
Yes.
See, the first one was about the aggressive phenotype of the hotspot mutation, but you answered already. And then she was asking also online on this conversation, what do you expect in cases of tumors with biallelic loss of function mutation? Because you nail it down that loss of function in one allele.
Yeah.
It's not pathogenic. Do you expect probably something?
So you mean in the, well, she probably means she's not here with voice, but if I understood well, the question is that if you find in a tumor 2 mutations, I mean, or loss of function or something. I don't know. I haven't looked into that, but probably if you lose the other copy, maybe the clone will just disappear, or I don't know. I have no idea. I haven't looked into it, whether because that's true. You may have the mutation and the other copy may have been lost by copy number alterations or another mutation, because this is highly mutated. But I don't know if somebody knows.
So if there are no other questions, I thank everybody again for attending. Thank you so much, Laura, for your presentation.
Thank you, and thank you for the nice bird chirping during the discussion. We could hear the birds, and thank you all for your attention and your questions. Thank you.